Um, Hi, everyone. Thanks for coming. Today for Z Evolution, we have Aimee Shultz giving a talk with some presenter friends that will introduce themselves as we get started. So thanks, everybody, for coming, and take it away, Aimee.
Awesome. Thank you, Sarah. Is everything looking good on the screen still?
Yep, we can see everything. Perfect.
Okay, so today we're going to be talking about a pipeline that we have developed within the lab that is allowing for this robust evaluation through evolutionary leverage of our gene models, or reelGene. And I'm Aimee. I am a fourth year PhD student in Ed Buckler's group at Cornell, and two postdocs, Shankai Xu and Brandon Monnier, will also be presenting with me today. And so in maize, we are really fortunate. We have, these 26 different NAM lines, as well as several other lines that have been sequenced at full reference level assemblies. But with that, we kind of have a little bit of a complicated nature where we have 1.5 million unique gene models across maize when we start looking at all these assemblies. And so as a maize geneticist, when we start wondering, oh, well, which genes do we want to study? And we start looking at all these gene models, the question now becomes, well, which ones are actually correct? And so if we look at just B73, the V5 reference genome, and we start asking, well, a correct gene should have all of the right components. So it should have a 5' UTR, as shown here, as well as our exons and our introns, and a 3' UTR. Around 75% of the B73 gene models look like this. Another 10% have all the components, but they are only a single exon. And then we have 15% that are missing one or more components of what we would consider a correct gene model. Now you might be wondering, okay, well, why don't just use the 85% that have the right components? But the real problem there is that we don't actually know if they're biologically correct. And so one way that we can address this is through evolution. And so we can theorize that correct gene models are going to have evolutionary support. So say we have one maize gene shown here, and there are 3 different potential transcripts. You'll see there's varying in the number of exons, as well as the actual splice sites for these different exons. And if we take 4 closely related other species and align them, we now can see that this 3rd transcript here is actually the one that is evolutionary supported. And so if we do this in maize, a great resource is this Anthropogonia tribe. And if we step out into that tribe, we actually can capture about 1 billion years of evolution that we can then use to better inform what makes a correct gene model. And so as part of the NSF PanAn grant, this was a huge collaborative effort with multiple different institutions. We've been able to go out and collect over 800 Anthropogonia species. This was done in part by Taylor Abushan-Elder and Toby Kellogg at the Danforth Center, as well as the 4 main institutions you see at the bottom of the screen. And all of the collaborators listed on the left-hand side played a large role in making this possible, both going out to the field and taking tissue samples, but also sampling from herbariums. And so by taking these herbarium and field specimens, we can leverage this evolution by sequencing and assembling the genomes of over 350 different grass species. So what I've done over the past year and a half or so is we've taken these herbarium and field specimens. We've gotten 30x Illumina short reads for whole genome sequencing. And then I've worked to assemble these genomes. And the goal, since they're short reads, is to just assemble the gene space. We're not really going to be covering these intergenic regions. And in the lab, we tried 3 different approaches. And the one that we ended up going with was this mega-hit metagenomics assembler. And so what we were able to find is that the short read assemblies can really capture the gene space. So shown here is a plot where we are evaluating around 5,600 genes that we would expect to be conserved across all of the grasses. And you can see that across these first 354 assemblies that I've completed, on average, we're hitting about 82% of these 5,600 genes are found as complete or duplicated. Another 17% are fragmented, and less than 1% are missing. And this was a bit of a complicated data set because around a third of these taxa are allopolyploid, and 20% of them were herbarium specimens, and some were over 100 years old. And so, back to this correct gene model question. We've gone from our short reads in the Androboganiae, and we've developed our gene space level assemblies. But what we really want to be able to do is pick the right gene models for our future genetic studies. And so really in our lab, we think a lot about, when we're thinking about what a right gene model is, the question becomes more along the lines of what's functional, what's producing a functional protein. When you think about the bioenergetic costs of a gene, making that mRNA is actually really cheap, but what's expensive is creating the protein. And so this problem becomes a little bit more challenging because we can't just go to the herbarium specimens and figure out, what proteins are there. So the question now has really become, how do we identify these functional genes and the correct gene models in species that we actually can't grow? And this is where reelGene comes in. It's this pipeline that's going to allow us to predict our functional gene models. And for the first step of this pipeline, I'm going to turn it over to Brandon.
All right, so now that we have all of these different assemblies, how do we start generating data that we start feeding into the model evaluation portion of this pipeline? But not only start generating data, but how do we do so in an efficient manner? So the first thing that we sought out to do was to evaluate all 72,000 B73 V5 transcripts. And so since we have all of these assemblies, what we did was we want to see not only if, but where these transcripts fall within each one of these different samples. And so for this first part of this analysis, we chose 91 of our assemblies. And so where are these assemblies coming from? So about 58 of these samples are coming from this initial Kew Botanic Garden release set. And this was essentially the short read assemblies that Aimee talked about in the first part of this presentation. And the other 33 samples are coming from this Pan Am long read assemblies project spearheaded by the Hufford Lab, and more importantly, Arun Seethram, who did a lot of great work generating these high quality long read assemblies. And so what we eventually did was we took each one of these transcripts and we mapped it to each one of these 91 assemblies using the Minimap2 alignment program. And so from this program, we're able to generate a series of metadata, namely where the transcripts are, or where they're located on either a contig or a scaffold, specifically where in that contig or scaffold it's located, and also the mapping quality for each one of these transcript alignments. And so what we can do is we can encapsulate that into sequence alignment mapping files or SAM files. So since we align this to 91 individual samples, we have 91 SAM files. And so what we're doing is we're passing this through a series of custom scripts that are leveraging a bioinformatics library called BioCollin, which is a library that was also developed in our lab. And what this is a high performance bioinformatics library that has syntax similar to Python, but has the compilation speed of Java. And specifically what the scripts or series of scripts is doing is it's filtering for high quality alignment. So primary alignments only. And so since we're trying to leverage evolutionary conservation, we're trying to have a strict coverage threshold across the CDS space for each one of these transcripts. So we have a greater than or equal to 90% CDS coverage for each one of these B73 transcripts. But in addition to that, we're also generating coordinate data for downstream parts of the pipeline, and then we're also exporting FASTA files for each one of these transcripts. So as you can imagine, we have 72,000 transcripts. This equates to 72,000 FASTA files. Each one of these FASTA files contains sequences for the respective transcript in each one of the taxa. And so what we do is we feed each one of these FASTA files into this multiple sequence aligner program known as MAFT, which is using a fast Fourier transform on the back end to help speed up the overall analysis. And so from that, we also generate 72,000 MSA samples. Each one of these has 91 taxa. And now what I'm going to do is I'm going to hand it over to Shen Kai, and he's going to talk a little bit about what he did with the junction portion for each one of these MSAs.
Okay, so now that we have the 72,000 B73 gene models aligned. So this will be our predictive variable of the model. So we're going to extract the features from the MSA and try to predict what is a good and what is a bad gene model. But before that, we need to first know what is good and what is bad for the gene model. So we decided to leverage the resource for very high quality assemblies and annotation of 26 diverse maze line, where we are able to distinguish the gene model into 4 different category: core genes that is found in all of the 26 lines, near core genes that is found in some of them but not all of them, and dispensable genes found in less than 23 lines, and private genes that is seen in only 1 of the 26 lines there. So from this, then we can classify all our 72 maze B73 gene models into different categories. So we can see that 72% of them are core and around 20% of them are dispensable and private. And we believe that if this gene is, or if this gene model is present in most or in all of the 26 lines, then they're most likely to be functional. But for those that is dispensable or even private, then they're likely to be the pseudogenes, which is actually not functional. Okay, so using this as a reference as our response variable in the model, then we will go first to look at the boundaries of different components of the gene model, which we call it junctions of the acceptor, splicing donor acceptor site, translation start and stop site, or transcription stop and start. So we will start from the MSA, extract 20 base pair junctions for each of the components, and then try to summarize them with some summary statistics. And then we aim to construct a XGBoost model to predict whether this junction looks like a good or bad junctions. Okay, so for those who are not familiar with XGBoost, it's one of the leading decision tree based machine learning models that is very suitable for classification problems. So we decided to use it. Okay. So a little bit more on how I extract the feature and try to summarize that. And since we have 6 different types of junctions, I will use the splicing donor junctions as an example. So this is a 20 base pair junctions of a splice sites. And we can see that with the red bar here, this is the GT motif for the donor. And from this junction, we are going to extract first the reference sequence motif, which is the splicing motif that is recognized by the splicing vectors. And we can see that although both positive and negative sets have this GT motif, but the signal is different in the magnitude. And in addition to the motif itself, we're also calculating some conservation estimates. First, we have the relative conservation score, or we call it phyto-P score. It is a measurement of how conserved the sequence is compared to the neutral sites of the gene model. And in this case, I use the foveal degenerative sites as the neutral model and check how are all those 20 sites conserved or not. And in addition to the relative conservation score, we also have the relative reference allele frequency measurements of all those 20 sites and the deletion frequency. And as we can see in all these plots, we can see a clear distinction of the patterns between positive and negative sets of junctions. Okay, so this is the features input for the XGBoost model. And next, we're going to see how the model performed. And we can see from the ROC curve and PR curves that both models have, for example, that for the splicing donor models, we can see a very high accuracy or relatively high accuracy and precision for the models. And if we extend it to all 6 models for different junctions, then we can see that all of them performs decently well in terms of accuracy and precision. Okay, so next, so what is the model actually learning? Again, we'll use the splicing donor example, and what I'm plotting here is the top 5 important features in the model. And we can see that for the first 3, it's phylo P score at site 11, reference sequence motif at site 11, and the frequency of the reference allele at site 12. And this is actually site 11 and 12 is actually the GT motif of a splicing donor. Okay, so we're using, oh, what am I doing? Okay, so we're checking, or the model is actually learning the GT motif and the phylo P difference here, or the reference allele frequency difference at the second site. Okay, so if we look at more all the models there, then we can see that the conservation at the splicing motifs or the start or stop codon itself are very important, as we will expect it. But we also notice that for the transcriptions start and stop site, the sequence completeness, or we call it the frequency of the deletion, may also be important there. Okay, and lastly, as a check of how of the performance of our model, we are checking how we apply the model to all the junctions that we have and compare what are the junction scores or what are the predicted scores of the junctions for the inner code transcript and the junctions in the dispensable or private junctions transcripts. And we can see that there's a clear distinction. Most of the junctions in the dispensable or private genes, they're actually not looking good evolutionarily. And this holds true when we applied it to all 6 different models there. Okay, so with the XGBoost model, we can evaluate how good the junction is. And this will serve as an input of our last part of the model, which Aimee will cover.
Awesome. So we've all gone through how we've done our MSA generation and the XGBoost model to get all of our scores for the junctions. And we know that junctions aren't the only important thing when it comes to gene models. And the actual components of the 5' and 3' UTR, as well as the CDS and introns, will contain information that could be very important for our model as well. So what we did next is we extracted several similar statistics on each of these different categories, and we fed those as well as the XGBoost model scores into a deep learning model called an LSTM, or long short-term memory model. If you're not really familiar with what this is, you could also think of it as kind of like the machine learning version of a hidden Markov model. And so from that, we get a transcript quality score of 0 to 1, which will ultimately tell us whether or not our model is predicting if this is a functionally conserved gene or not. And so just to walk you through exactly what our inputs are looking like. So for a single transcript, there's going to be an input matrix, and the actual length of this transcript is going to vary depending upon the length of the gene model. So you can see here that there are 5 components relating to each of the different major components in our example transcript. And what's fed in first is various estimates of sequence conservation. Next, we take in different features that involve how fragmented the alignment is, as well as estimates of selection constraint. And then we also have our junction scores. And you'll see that, you know, the different NAs in here will allow us to do an additional step of almost kind of a one-hot encoding. So we're telling the model, and it's going to be learning on whether or not it's a leader sequence, coding, intronic, or a terminator sequence as well. And so with this, we train the model over 10 iterations using a leave one chromosome out approach. And so what happens is that in our first fold, our training and validation split occurs in chromosomes 1 through 9. And after the model is completely trained, we then run chromosome 10 through the model and get a series of predictions with scores ranging between 0 and 1 for that chromosome. This is then replaced. The model's restarted and rerun with the second fold, now inputting chromosomes 1 through 8 and 10. And then we're testing it and getting values out on chromosome 9. This is repeated all the way till we get to fold 10, where we are getting our test values out for chromosome 1. And so overall, our model is showing high precision and accuracy. I'm showing here the ROC and PR curves for one of the folds, but they are similar across the board. And you can see that, it's doing relatively well here. And when we start diving a little bit more into the biology here of what we're seeing, only the core genes are largely predicted as functional by our model. So what you're seeing are just the B73 genes with our best reelGene transcript. So think of it as the representative transcript for each gene. So we're hitting 78% of our core genes are predicted functional, 44% of our near core genes, and then 5% and 2% of our dispensable and private genes are being predicted as functional. And one thing that, we're thinking could be of interest here is that you could actually start diving into maybe, those genes and seeing why are they predicted functional, what's making them so conserved. And these potentially could be sources of adaptation or other traits of interest that people could look into. Now, when you look at all 72,000 transcripts, you see relatively similar numbers across the board here for these 4 different classes. Now, you might also be wondering, okay, Aimee, well, you train this model on core versus dispensable and private. How does this actually relate to transcriptome or to the proteome? And so reelGene actually correlates with the expression and genes in the proteome. So shown here is our best reelGene transcript for each gene. And you can see when we use Washburn et al.'s transcriptome set that largely the transcribed genes are being scored very well versus the not transcribed genes are being scored poorly, as well as when we look in the Wally proteome set, we're seeing the translation is scoring very well versus not translation. And if we look at the AED scores, which is another metric of just estimating basically how correct might these gene models actually be, we do get a significant Spearman's rank correlation of -0.39, which the lower the AED score, the better. And so if we look at all transcripts, so this is all 72,000 gene models and B73, we see that this trend still continues. And now, as a little bit more of a diving into some genes that we would expect to be real, I pulled the Maze classical gene set, and 92% of our best reelGene scoring transcripts for each of the Maze classical genes are being predicted as functional. So this is showing that, while this is kind of our initial run through of this pipeline, it is proving to be very promising as we move forward. And so what's next? Well, we've developed reelGene scores on all B73, B5 gene models, but we want to expand this out to all B73 gene models. So thinking going back to V2, V3, V4, and comparing the scores there. We also want to do this in the process of running it on all NAM gene models. And we will be generating these scores on all of the brand new Pan-Am gene models. And ultimately, our goal for this pipeline is for it to not only be a community resource of just scores that you can go and pull on all these different gene models, but also a pipeline that you can use to evaluate your own gene models. Say there's a different species other than maize you want to look at. And we ultimately will be doing this again with all the Pan-Am assemblies. So not just our subset of Q short reads, but the plan is to include, all the short reads and all of the soon to be released Pan-Am genomes. And so I hope today we were able to really show all of you this new roadmap for how we can uncover our better gene models and find functional genes using the Anthropogonii, going from short reads and gene space level assemblies to our machine learning model, which again will pull out our best gene models that are most likely to be functional and hopefully can help clarify and improve all of our future genetic studies. And so at this point, there's a whole host of people that we need to thank within the Buckler Lab, as well as the Pan-Am grant. We'd also like to thank our funding sources, and we hope you'll keep an eye out on bioRxiv for a reelGene manuscript coming soon. So at this point, we are happy to take any questions.
This is so exciting. I am excited to see how what genes show up in the reelGene lists. It looks like Matt has a comment. Do you want to ask a question?
Yeah. So it also seems kind of expected to me, Aimee, that you would see the core, like the model when it's trained on the core and the near core genes, that you would see expression and translation of those genes, because you're typically going to see those type of genes more constitutively expressed across tissues and also more highly expressed. So it seems like those would pick those up. I'm wondering, I mean, do you think that if we focus on models that look like those core and near core genes, are we going to miss real genes that are unique to particular species or potentially more important for adaptations across different environments within species? Is that just kind of a trade-off that we'll have to kind of be okay with if we want to try to enrich for genes that are more accurate in terms of their models?
Yeah, so with this, we left out the near core completely in that training set because we were kind of curious a little bit about that same question that you just posed. Like, they could, they probably look more like core genes, but what's going on there? You can see even with the near core, there was a bit of that split. I guess yes, we are going to have to weigh, that question you just posed where, okay, is it going to be more biased? But also the part that I find promising is that we are still pulling out in the private indispensable genes, some that do show, what some of those strong signals are. So for where there are alignments, are we seeing that they are correct? We're getting the correct splicing motif we'd expect to find. Are we getting the correct signatures, of an exon within there? So it's looking at each of those specific components and not necessarily how many different species does it align to, if that makes sense.
Yeah, I think that was an exciting aspect too, that you're picking up some of those genes. I think it'd be really nice to follow up on those, maybe for your publication, take a look at those genes and, see what they're doing, see why they look, like they're picked up versus the other genes. And then the other question that I had is just curious, is there a way, with this pipeline that you've developed, to not only identify what we think are the real genes and the good gene models, but also to improve the existing models for, what might be real genes, but where, we might just have an incorrect annotation of the model?
Yeah, so we haven't looked into that yet because kind of the main goal we've been saying is that we're not trying to like annotate new genes. We're trying to just find, what is a correct versus incorrect annotation. That being said, we can, and there could be a potential, we haven't done this yet, but to actually go in and look at what was being scored where, and maybe you could point out like a certain exon or a certain junction was scored very poorly for this particular gene model, while the other exons and junctions were correct. So that could maybe indicate there could be something going on, with the junction.
Constraint across, the phylogeny or even within species at a particular junction, and then like CML 333 is, wrong at that position.
Right.
It seems like there could be a way to fix that, Cool. Yeah, really neat work. And yeah, it was fun to hear you talk. I'll let other people ask questions.
Hi, Aimee. I had a quick follow-up question on the first question Matt asked. Yeah, most of the features that your model is being trained on are, I would say, the features of a well-established gene, right? Like, but what about the genes that are, what about young genes that are on their way to being established? Like, how are they accounted for? Will it be not better if you also consider some of the features such as chromosome accessibility, like translation, riboseq, like translation ability, and those kind of features as well for training your models? Even the proteomics data support for some of the gene models, or maybe AlphaFold support as well. Those kind of things might help. I think. What do you say?
Yeah, I guess. Shankai, do you have any thoughts on
Yeah, I think I understand what you mean, but do you mean that like we can try to fold the CDS of the reference gene model and see if it's foldable or if the folded structure makes sense or not, and use it as another features in the model, right? Yeah, but
Yeah, could be something we could
yeah.
add.
So if I could jump in real quick, so the thing with AlphaFold is it's fine if some of these fold, but it doesn't really do well on these things that are unstructured by
I know this so
virtue.
it's based on the alignments, but there's like there's a fork of AlphaFold that works for the orphan genes that's able to predict non-conserved proteins as well, like for specifically for the young genes. So I was thinking more of that. I couldn't remember the name, so that's why I said AlphaFold. Sorry.
Yeah, I think orphan genes, you're absolutely right, but I mean things that are more just intrinsically disordered. So it could be a real gene that doesn't have a defined 3D structure, and it just can't. You wouldn't be able to get those.
Thank you. Great.
My question is, how do you envision implementing this? Is your vision that you'll have an improved annotation set, or is your vision that you'll annotate the annotation to say which ones are more likely real genes?
Yeah, so at least right now, the first thought is we can just release all the scores for everything we've run through the models, so people can go and say, you know, I'm interested in this gene and I'd like to pick out which transcript I should focus on. Or is this gene even correctly annotated at all? And should I go to an older version? You know, that type of question. So releasing all of those. But then there's also, we're discussing in the lab how we're going to actually implement the pipeline and make it so that if someone has a gene that we don't have a score on, they can actually run the pipeline themselves and generate their own scores.
Great. Yeah, I'm always hesitant to hear if people want to like reduce the size of the gene set, because, you know, even with the you know, more real genes, a lot of them still they are transcribed or they aren't transcribed, but they're more likely translated, right? So
And so
yeah,
that's where the thought again is like, we're not saying, no, oh, let's reduce our current gene set. It's more, this is another tool in the toolbox that people can use when trying to make a decision on which, you know, transcripts do they use versus, you know, just throwing all of them in there in their analysis.
Yep. Love that.
Yeah, I mean, maybe for the maze assemblies that are on MazeGB, you know, you could add a column to the GFF file that's the reelGene score, and then people could just filter, you know, based on the reelGene score if they want to, if they've got some sort of threshold that they'd like to.
I was gonna suggest that because James Novel they released the new function for most of the gene models that were only uncharacterized protein in our set. So you could add another column there. Adding these values would be really informative. Thank you.
You don't even need to filter. You could just use it to, you know, check how real reelGene says it is.
Hey, Amy, that's really cool work, and the talk was really nice as well. So, congrats to all three of you on this project. I'm curious, do you have a feeling for how conserved is the biology that this model is learning, or like to put it another way, how far outside of the Andropogonii do you think it can extrapolate? So thinking about crops or other species with less resources to annotate gene models, you know, could it be applied right out of the box, or do you think we need to be careful about, you know, extrapolating outside the training space?
Yeah, so that's actually a question I kind of want to address in the paper, asking, because the part that's exciting to us about this is that, you know, we're leveraging short reads. And so in theory, someone could implement this model from scratch, so not using our fully trained data set, essentially, putting in their own MSAs, basically, and run the model that way. So they're able to go through, say, they have a whole set of short reads for their species that are closely related. They could run the model that way. I don't want to say, like, how about we could extrapolate out the core versus non-core classification we have, say, going out to wheat or something similar. I don't have a good feel for that. I don't know if Shankai, if you have a better
Yeah, I think
thought or.
my opinion on this is that we're training based on a biology that is diverge for like at most 20%. And if we go beyond that, then maybe the model wouldn't be suitable. But if there's anything like any other pan-genome paper or project that is going on, maybe this will be useful.
Yeah, and they could essentially take the same architecture and generate their own version of the scores.
Yeah, I guess I'm thinking about application for species that don't have the resources to generate pangenomes or train up their own models, but you know, like other grasses that are maybe closely related. Or I am trying to think if there are other you know, I mean, you could go crazy and see how well you can predict, you know, gene models in like Arabidopsis or something, as a test of like whether it can work in really far-flung cases. I don't know that I have anything in mind that's more closely related that you could test on that you know, could feel confident in, but might be nice to know.
Yeah, that's a good suggestion. We'll see if we can give that a try. I think Ruben, do you have a question?
Yeah, hey, Amy, Brandon Shankai, thanks. I guess this is another twist on what Joe was saying now and what Matt mentioned before. I wonder if you have an intuition of how your results would change if you would use a larger collection of assemblies from other species to build your models.
Yeah, I think my gut feeling here is that what's more important than just sheer numbers is going to be actually how much of the phylogenetic tree are you covering, in the sense of, you know, getting our conservation estimates. I think it would be better to have a wider array of samples, maybe covering, you know, different areas of the tree versus if we're just strictly doing, like, say, 100 samples that are only in one very specific area, depending on like how broad you're trying to go with your sample, right? There's like pros and cons. So I guess we haven't played around with the numbers yet or looked at, you know, how diverse, you know, how much of the tree we're covering and kind of what that threshold is. Again, that's something I want to explore for the paper coming up.
Cool, thanks.
One more comment too. I mean, I wonder if it's possible down the road to incorporate into the model the level of conservation that you see within species. So, you know, we're going to be moving into a, you know, multiple assembly per species world. And, you know, if you could somehow penalize conservation a little bit, you know, across species, when you actually see conservation within species, maybe we'll be able to pick up some of these real genes that exist within species. I mean, because we know we've got rapid evolution that occurs along lineages and, you know, neofunctionalization of real genes and genes that organize and, you know, are species specific. So I wonder if
we definitely have found Zia-specific genes through this whole process, and something we could do, we didn't for this talk, but actually pull out and see where the Zia-specifics are scoring.
Yeah.
Kind of see if that answers a bit more of your question.
Yeah, and if there's some way to tailor the model when you've got multiple assemblies per species and you want to be able to capture those species-specific genes. And not penalize them based on constraint across the phylogeny.
Now that you mentioned that, Aimee, do you see sub-SIA specific conserved genes?
Say that one more time.
So you mentioned that you have identified SIA specific core genes. Do you see different SIA species core genes as well?
Oh, okay. So we have not looked into that, but that's something we also could do and start splitting out based on, you know, these MSAs that we have.
Right. Obviously, we're thinking
Interesting, like if there's, yeah.
and the diplo. Yeah.
That'll be something too, as we dive into actually running all of the brand new Pan and gene models. Once we run those in as well, we can really get at, are we seeing, you know, these core ZIA specific genes, and do we start seeing, you know, different taxa within ZIA, them dropping off one or another, that type of question. So that'll be something we absolutely can get at.
Yeah, going back to what Matt said at the beginning, I keep thinking about that. Whether you could find a more environment-specific core genes that you could identify across different lineages. Right. Something obviously like cold versus hot environments and things like that.
That would be super cool. Kind of an
Yeah, I'd have
environmental
to think about how we would do that other than actually splitting up our alignments so that, you know, we're really just honing in on environmentally, okay, these are in a similar region, let's run them through and see what their alignment scores are. Something like that could be done as well. Again, right now, this pipeline that we have is more aimed to just be broad and encompass, you know, all 72,000 gene models that we have, and not really trying to look out for, you know, is this just found in one, you know, is this a real species specific gene, or is this environment specific gene kind of thing. But definitely I could see with the MSAs finding a way to leverage that.
Well, obviously you're doing something really cool here, Aimee, because, you know, it's generating lots of ideas of how you could extend it. So you should take this as a compliment. That
oh no, we're taking notes in here. We're very excited.
Yeah, I just wanted to say this is super exciting to see this whole thing together. You know, following the discussion on Pan Am is a lot of fun, but seeing the big picture has been great. And, you know, I think even if you only quote unquote, pull out the core genes out of all the Andropogoni, that's going to be a giant contribution right there. And yeah, all the orphan genes or the lineage specific genes is going to be cool, and I'd expect that, but you know, I think that's going to happen. But just sort of the basic focus on the core genes is a really giant step forward. So it's just been fun to listen to.
Awesome. Thank you, Toby.
Yes. It's
Any
really
other
exciting.
questions?
Any other? Yeah. You don't need me.
Sorry, Sarah.
No, it's okay. I was trying to moderate, do my job. All right. I don't see any more questions. Exciting work. Thank you so much for presenting. I'm excited to see the paper, and thanks everybody for coming, and we'll see you next week.